The market squid is perennially in the top three commercial fisheries in our state, the other two being the sardine and the sea urchin. Market squid breed close to shore and form massive swarms that can be easily located and exploited. Moreover, it's a sustainable ocean harvest. Commercial fishing for dory tooths is pretty carefully regulated, and it has very little off-target bycatch, and the growth rate and reproductive capacity of the squids are great. And they are very tasty. Anyone who has enjoyed calamari rings at a local restaurant can attest to this. At this point, I'd like you to take your squid and first determine if it, and squids in general, belong in the octopodiformes or the decapodiformes. Okay, so for full disclosure, I know I said I'd be dissecting a market squid, but I ended up buying a different species of squid from a Ranch 99 market instead. I think it's an Ilex illicebrosus, the North Atlantic short-fin squid, considerably bigger than a market squid, and so it will actually be a bit nicer for the visuals. Right now, though, you're looking at a Doryteuthis market squid, but note that I'll be switching back and forth between this and a darker colored Ilex squid through the video. Most of the anatomy is basically the same anyways. And yeah, so there's a head with all of the arms and tentacles, and the mantle, which is the main tubular part of the squid's body. If you are looking at an actual animal in front of you, there's a possibility that it lost a leg or two, or like in this one, two of its legs are stuck together. But a complete squid has 8 arms that are shorter, as well as 2 longer tentacles, for a total of 10 appendages. Therefore, if you said decapodiformes, congratulations. Give yourself a slimy pat on the back. Each arm and each tentacle has dozens of small suckers, and in my ilex squid, each sucker is equipped with a hard keratinized ring with tiny but sharp teeth. These help the squid suckers get a stronger grip on their prey, and this is also the reason that squid suckers will sometimes leave a mark on your skin. The smaller market squid don't have these rings. In some, if not all cephalopods, each individual sucker is controlled by its own motor neuron, going from the animal's brain out to each of the hundreds of suckers. The 2 longer tentacles have great reach and are also retractable, so the animal is able to use them to reach out and ensnare a prey item, maybe a little fish, and then pull itself closer to the prey, and then grabbing hold of the arms, it can start to eat. At the center of the arms is the mouth. There's a ball-shaped buccal mass that provides the musculature putting force behind the squid's bite, for which it uses this hard keratinized beak. It actually resembles a parrot's beak with pointy ends and sharp scissor-like edges that let the squid bite off one little chunk of flesh after another. The market squid is little, and yes, it is a delicious morsel to many larger predators. But even the little market squid is a pretty formidable predator on its own, and if you give it a chance, it will give you a painful bite and leave a bloody little divot in your skin. If you reach in with your fingertips and pull on the buccal mass, it'll pop out, and you'll see the esophagus that connects the mouth with the rest of the GI tract that leads to the stomach that sits partway up in the mantle cavity. Now don't cut anything open just yet. We still need to cover the rest of the external anatomy. You'll note that your squid has a nice big eye on each side of the head. Not surprising for a visual predator that also relies on vision to detect and escape from their own predators. They also use visual signals to communicate with other individuals of their own species. These signals are transmitted from the spots you see on all the surfaces of the squid's body. Look really closely and you'll see that these spots are actually small liquid sacs. These are the chromatophores. Zoom in on a patch of skin on a live squid and you can see how the little sacs of liquid pigment expand and shrink, causing the skin to change color. The most abundant pigment on our squid skin is melanin, the same pigment in human hair and skin. By making the dark chromatophores wide and flat, that is, covering a larger surface area, the squid will appear nearly all dark brown. By shrinking down the chromatophores narrow and tight so that each is a tiny pinprick of color on an otherwise white mantle, the squid can make itself turn ghastly white. Presumably, the squid uses these and also more subtle signals involving secondary chromatophores carrying the other more colorful pigments, the blues, the greens, the yellows, the reds, in its communications with other squids. Some species of octopods have spectacular control over the chromatophores, evident by the way that they can mimic their surroundings, practically blending in with whatever background they nestle up against, a useful trick for sit and wait predators. This also requires some pretty sophisticated brain power, an ability to detect and reproduce a pattern both in color and texture. This, together with fine-scale motor control over chromatophores and suckers, not to mention the complex musculature of the arms, all make a strong case that cephalopods, and octopuses in particular, have a high degree of neural sophistication, the Einsteins of the invertebrate world. If you place the squid ventral side up, you can see that attached to the head is the siphon, which is the smaller end of a funnel whose wide top sits usually just inside the opening of the mantle. At the tip of the siphon is an even smaller opening. Squids use jet propulsion to swim. Pressurized water from the mantle cavity gets captured by the funnel and sent down the siphon. The opening at the tip of the siphon is controlled to direct the flow of water. Water inside the mantle cavity is pressurized by contraction of the mantle musculature. The collar part of the mantle, that is, the opening, narrows, pressing against the top of the funnel, the wide side, and so the only way for water to leave the mantle cavity is down the funnel and through the siphon. Here's where a little physics comes in handy. The speed of flow of a moving fluid is inversely proportional to the cross-sectional area of the tube that it flows through. By capturing a large volume of water at the top of the funnel and pushing it through a very narrow siphon tip, the squid is able to produce a super fast jet of water, which means that the squid can attain higher speeds and faster acceleration. By controlling the direction that the tip of the siphon is pointing, the squid controls its movement. With the siphon opening pointing straight back, the squid motors mantle first, taking advantage of its torpedo-like hydrodynamic shape. By turning the siphon to point toward the pointy end of the mantle, the squid moves head first, as when it would need to attack a prey or a mate. We'll get to that soon enough. The squid also has fins near the tip of the mantle. These can also provide some propulsion by pushing water forwards or backwards, and they offer tracking stability. You've probably noticed that the belly of the squid is lighter in color than the back. Chromatophores on the ventral side are fewer and or smaller compared to those on the dorsal side. This pattern of light underneath, dark on top is called countershading, and it is super common in animals generally. The idea is to show white or silvery to any visual predators that are below you in the water. The surface of the water is usually bright during the daylight, and so seen from below, you would blend in with the lightness of the sky. Meanwhile, dark coloration on the back or dorsal side means that predators looking down at you from above will have a harder time seeing you against the darker background, as with the ocean depths. In a certain way, countershading is the squid's attempt at becoming invisible to its predators as well as its prey. Another thing worth noting while we're still pondering the squid from the outside is that the hydrodynamic torpedo-like shape of the squid's mantle is made more effective because it's relatively rigid. This stiffness of the squid's mantle, which is important because it allows the squid to move faster as it escapes from predators, is due in large part to the pen, which we'll remove next. On the dorsal side of the mantle opening, the collar comes to a point. If you pinch firmly, I use my thumbnail to do this, and pull, out comes this plasticky insert in the squid's mantle. I only got half the pen from this Ilex squid that I bought. The front half of the pen is still in the mantle. The squid's pen is analogous to the notochord that we'll eventually be talking about in our chordate discussions. And the general idea is that sometimes just adding some stiffness, not too much, actually improves your ability to swim. The squid has its pen, while the cuttlefish, another decapotiform, has its cuttlebone. Both are remnants of what was once a robust shell of their coleoid ancestors. Octopuses, too, were once shelled cephalopods that, through their evolutionary changes, came to lose their shells. Entirely in the case of octopuses, and nearly entirely in the case of squids. Why would evolution make squids lose their shells, which presumably offered them some protection against predators? What advantage did squids gain by unburdening themselves of the heavy shells carried by their ancestors? Why, mobility, of course. We'll actually see the same theme next week when we consider the evolution of fishes. The earliest ones had heavy body armor, and then, over time, fishes took on more lightweight skeletons and lightweight scales. Before moving on to the things that are on the inside of the squid's mantle cavity, let's review the basic anatomy that we've covered so far. Arms, tentacles, and their suckers. Mouth consisting of a buccal mass and beaks, and an esophagus that carries food toward the stomach. Chromatophores, siphon and funnel, and fins, and you want to know how the squid uses these to move around, and the pen. The next thing to do will be to make an incision midline on the ventral side of the mantle, cutting all the way up from the opening of the mantle at the funnel to the tip. Scissors are good for this kind of cut because you have less of a tendency to cut through any of the structures in the mantle cavity. Now, the first thing we'll do is to distinguish boy squids from girl squids. The gonads can actually get in the way of our seeing the other parts of the squid's anatomy, especially during the breeding season when they can occupy a large fraction of the mantle cavity. First, we'll figure out the sexes, and then, if necessary, we can get the gonads out of the way and move onward with our dissection. Females, and this is a female shown here, have large, firm, translucent ovaries and opaque white nidamental organs. Males, on the other hand, will have a single testis that's soft and gushy and uniformly milky colored, and another sac that looks like it contains curdled milk. This is the spermatophoric sac, containing the squid's spermatophores. Spermatophores are little packets of sperm that the male prepares and gives to his mate, much like a box of chocolates on Valentine's Day. This is actually pretty common in invertebrates. In the case of market squids, the male has a short penis, and so it needs to use a tentacle to take spermatophores from its own mantle cavity and then stuff them into the female's mantle cavity and hopefully to the right location where the sperm will be taken up and used to fertilize the eggs. The eggs, once fertilized, get packaged into egg sacs containing up to 200 fertilized eggs, and then the female squid attaches her sac of eggs to the sea bottom. A female market squid can produce up to 20 such egg sacs in her year of life, and then dies. Yes, they're like annual plants, living for only 1 year. The babies emerge looking undeniably adorable and start eating immediately, and before long we have the next generation of market squids. Now, if the gonads were in the way, you can remove them. If not, you can leave them in. The next thing we'll consider is the squid's circulation. The first thing to do is to locate the gills, also called ctenidia, with a silent C at the start. These are soft, feathery things that attach to the inside of the mantle on either side of the main visceral mass. Circulation in the squid, as well as all cephalopods, is closed, meaning that blood is pumped by the hearts and remains within vessels throughout the entire path of its circulation. There are actually three hearts doing this pumping, but these hearts aren't exactly easy to see. You can find them by knowing what to look for and looking in the right place. Each of the gills has its own branchial heart. That's two gills and two branchial hearts, two out of the three hearts total. The branchial heart is at the base of the gill and pumps oxygen poor blood out to the gills where gas exchange with water in the mantle cavity occurs. O2 rich blood returns back from the gills to the systemic or aortic heart, which then pressurizes the O2 rich blood for moving it out to the head, the mantle, and the visceral organs. There's an analogy between what we have here and the double circulation in our own closed cardiovascular system. We also pressurize the O2 poor blood to send it out to the places where gas exchange occurs. But instead of using two branchial hearts, we have a right ventricle. We also pressurize the O2 rich blood returning from the lungs for moving it out to the systemic circulation. But instead of using an entirely separate heart like the squid does, we have a left ventricle on the other side of a septum separating it from the right ventricle. Not all animals have a heart pressurizing the O2 rich blood after it gets oxygen from the respiratory organs. Fish, for example, push blood through the blood vessels of their gills, but then they depend on residual pressure along with muscular contractions of the body to keep the blood moving along after it leaves the gills. Now, we won't be tracing the GI tract of the squid. We saw the beginning earlier with the mouth and the esophagus, and now we'll just locate the general area where the anus is. Now, you may have already noticed the ink sac. It looks like a thin, silvery tube that stands out color-wise from the otherwise drab color of the squid's innards. The ink sac contains a highly concentrated melanin-rich mixture. When the squid wants to confuse a predator, it just has to let a small dose of ink out into the water that it squirts out from its siphon, and the resulting cloud will hopefully be enough to distract the predator, allowing the squid to escape. Well, this ink sac's opening is near the top of the funnel, right? Well, that's also where the anus is, and this makes perfect sense. If you're going to have to poop inside your mantle cavity, at least do it where the poop will be ejected from the mantle cavity with the next jet of water that you push out through your siphon. Next, we'll address something of importance in the history of biology and specifically neurobiology. Many of you will have some general awareness of how neurons work, the way that they use membrane channels and ion movement to produce an action potential that gets propagated down a long axon, allowing one part of the body, like the brain, to communicate directly with the parts of the body that are sometimes quite distant. Well, squids figure significantly into much of our early learning about neurons. Why? Well, they, like a lot of invertebrates, are in possession of supersized nerve cells, cells that are big enough to manipulate with the insertion of patch clamps to measure the voltage differences across the membrane of a living cell. These giant axons require some explanation for their own sake. Now, what's up with these giant axons on giant neurons? We just got through saying that squids and octopuses have huge neural complexities with fabulously high numbers of neurons for just the motor control of individual suckers and chromatophores. Take that, plus the octopus's ability to solve complex problems, and the fact that this animal's brain is not the size of a watermelon tells us that most of the billions of neurons must be normal sized. That is, long, but extremely narrow and generally very small. Why would there be neurons of this extra large variety? Again, there's physics, this time biophysics. It turns out that the speed of an action potential down the length of an axon is directly proportional to its cross-sectional area. A thicker axon moves information incrementally faster than a thinner one. In the case of a giant axon, the effective motor function is the generation of a powerful contraction of the mantle musculature, for example, allowing the squid to escape from a predator. The squid with a slightly thicker axon responds a bit faster and is less likely to get eaten, more likely to survive, having a higher fitness relative to the squid with a thinner axon. It's natural selection that has led to this extreme size and specifically those neurons that carry signals for running away from predators. If you look near the collar of the mantle, where the visceral mass attaches to the dorsal side of the mantle, on either side, you'll probably have to nudge the visceral mass slightly to the right or left with your finger. You'll find a little star-shaped ganglion, the stellate ganglion, which is a relay connection between the brain and the innervation of the mantle musculature. That's a little piece of history right there. By the way, you might remember from another biology class something about how our nerve cells make nerve signals move down the axon very, very rapidly. Myelin sheaths and saltatory conduction. Now that's a purely vertebrate phenomenon. Invertebrates, including squids, cockroaches, and sea slugs, all lack myelin sheaths, and so they're reliant on giant axons for those speedy responses that are most important for their survival. As a final task, I'd like you to consider the squid's eyes, which bear more than just a superficial resemblance to your own eyes. The basic parts are the same. A transparent cornea that keeps the water from moving in and out of the vitreous space, an iris, a clear ball-shaped lens, and a retina composed of photoreceptors. This part-for-part correspondence of eye anatomy between vertebrates and cephalopods is a classic example of convergence. Two lineages that through independent evolutionary paths find effectively identical solutions to meet the need of acquiring visual information from their environments. If you like, you can open the squid's eyes. But just a warning, this part will be messy because the pigments of the photoreceptors will leak out, but you'll be able to find the lens, which will look like a small transparent sphere. To recap the anatomy we've seen inside the mantle cavity, there were the reproductive organs, ovaries and nidamental glands in females, and testis and spermatophoric sac in males. The cardiovascular respiratory anatomy, two gills, or tonidia, each having its own branchial heart, as well as the systemic or aortic heart. Ink sac, stellate ganglion with a giant axon, and some eye anatomy such as cornea, lens, and retina. In the next part of our laboratory video, we'll be opening up a live mussel and looking at the anatomy inside its mantle cavity. If you want to do a dissect-along, you'll need to find a small live mussel. We collect ours from the docks in Oceanside Harbor. You'll also need a supply of cold seawater to submerge the mussel in as we make our observations.